The evolution of populations. So when you define a population, remember the definition, the first thing, it's a group of the same species in a given area. And the same species should be able to interbreed and produce viable offspring. Viable means that the offspring are healthy and also able to have other offspring. Remember the sentence in bold, a population is the smallest biological unit that can evolve. So you, as an individual person, you are not evolving, okay? The smallest unit that evolves is a population. So the population of, and remember populations can be very, you can just think of whatever population, like population of Los Angeles, right? That population can evolve over time, but not you yourself. So individuals don't evolve anymore. Or they've never evolved. The definition of the evolution deals with populations. Anyhow, so we're going to look at gene pools and the mechanisms of microevolution and macroevolution. So to understand a gene pool, right, it's a collection, total collection of alleles in a population. So let's go with, the population of humans in Los Angeles. We can look, every single person's alleles that they have, we have 2 alleles for every trait, and for every trait that we have, so that's our gene pool. So gene pools can change over time, okay, and that's basically what microevolution is the change of gene pools over time. It's a change of gene pools and it's a change of allele frequencies over time. We'll look at that, but let's take a look at what's on the screen. So how can you actually make a genetically variable individual in a population? So how can you make a different individual? And very simply, probably you're thinking of just 2 separate people having an offspring. That offspring, their child, will be different than them. So that's number 2, right? Sexual recombination. So if you are an organism that sexually reproduces, you're always going to have a genetically new offspring, so that increases variation in a population. But the second thing is mutations, right? And this is less thought of and less common, but mutations happen and they can actually make changes in alleles, so that can also introduce some variation into a gene pool. Okay, so let's analyze gene pools. So when you analyze gene pools, you look at allele frequency. So how frequent is an allele is basically this, how common an allele is found in a population. So let's think about, I know this is not true to life, but let's just think about blonde hair versus brown hair. And let's think about the gene pools of maybe Swedish people and the gene pools of people maybe in China, right? So clearly, how frequently are you going to have the blonde allele show up in the gene pool of Swedish people, of Sweden, right? You're going to have a lot more blonde alleles there than you are in China. China's probably 0 blonde alleles, right? So there's no blonde people ever for the history of China, I don't think, unless they dye their hair. So the frequency of that blonde allele is much higher in Sweden than the frequency of that blonde allele in China. Opposite, you can think about the brown hair allele, right? The brown hair allele is going to be much, much more frequent in China, almost 100%. And then if you look at Sweden, there are dark-haired Swedish people, right? So the frequency of how often do you see this allele, this brown hair allele, is probably not going to be as much as China. So it's a lower frequency of brown hair in Sweden. Anyway, so let's look at this example. So there's an insect population that there's a gene, okay? So we're talking about a gene, and the gene codes for an enzyme that breaks down a pesticide. Allele A is a functioning enzyme. Allele a is a mutated enzyme, so this is non-functional, okay? So these are 2 alleles. Remember, you always have 2 alleles for every gene always, because you have in an individual, right? You have 2 alleles in an individual. In this insect population where they spray a pesticide, which allele do you think will have a higher frequency? Okay, so hopefully you should think to yourself, if they spray the pesticide, the surviving individuals would have to have broken down that pesticide, right? So they have to have the functioning enzyme just to live. So in that population of insects, if they spray that pesticide and they constantly spray that pesticide, just like that example of the cockroaches we made the other day, or yeah, in the other lecture, if they constantly spray that pesticide, then you expect this allele, allele A, with the functioning enzyme to be more prevalent, to be more frequent than the mutated allele. The mutated allele, since it's recessive, it could be in that population, but it, you know, as a heterozygous individual, you could have be big A, little a, and survive the pesticide. So the little a frequency won't be 0, but it'll be much, much less than the big A, all right? So we're going to look at allele frequencies. So this is going to be a little scary, but you're not going to have to do any math here. So this is just recognize this Hardy-Weinberg formula where p squared plus 2pq plus q squared equals 1. So this is genotype frequency. So so when you remember we said big A, little a, so these are the two different alleles for the same gene. So in this example, the p squared is the homozygotes of the dominant allele. PQ, this is the heterozygotes, right? So you can say it's big A, little a. And then Q squared would be the frequency of homozygotes on the other allele. So like little a, right? When you put the allele frequency, if you get the allele frequencies as numbers, which you can, and you plug them into this formula and it equals 1, that means that the gene pool is stable and not evolving. So a non-evolving population is in genetic equilibrium, also known as the Hardy-Weinberg equilibrium, meaning the population's gene pool is constant over time. All right, so what you want to take away from this slide is that there is something called the Hardy-Weinberg formula. You should know what these stand for, and that if you plug in the allele frequencies into this equation and it equals 1, then we have a non-evolving population, and that, another way of saying that, it's in Hardy-Weinberg equilibrium. Okay, so it's not evolving, there's no changes to that gene pool, all the allele frequencies are the same over time. It's a little theoretical, that doesn't really happen, but let's look at this. So this is something I would actually expect you guys to be able to do, is to plug in numbers while you do a Punnett square and figure out the allele frequency for different genotypes. So these are going to be given to you guys. You're going to know the P and the Q. And let me write down for you, P plus R equals 1. So whenever you get a problem like this in a test or in a quiz, you're always going to be given at least one of the alleles, right? So here we're looking at big R, little r. The big R stands for red flowers, the little r stands for white flowers. And maybe I'll tell you that the frequency of big R is 0.8, right? So if I say, okay, the frequency of big R is 0.8, then you should know what the other one is because it always equals 1. So 0.8 plus 0.2 equals 1. Okay, so you know this. So I will definitely give you one of these allele frequencies, but you need to figure out the next one, and you do that with this formula. So when you do a Punnett square, so if I said, okay, here's your allele frequencies, we're going to mate 2 heterozygote individuals. So PQ, PQ, but in this case it's big R, little r, right? So you just put in the numbers. So you see there's 0.8. You put in the numbers here along with the allele frequencies, right? You put them in next to your letters. And then when you fill in the boxes, you multiply the numbers. Okay, so it's 0.8 times 0.8 is 0.64, 0.2 times 0.8 is 0.16, 0.2 times 0.18, sorry, 0.8 is 0.16, and then here 0.2 times 0.2 is 0.04. So now you have the allele frequencies of the different genotypes. So if I said, hey, out of this mating, what is the allele frequency for homozygous red? right? What's the allele frequency for homozygous red? What's 0.64? So another way of looking at this is like 64% of the offspring are going to be big R, big R. That's the probability, okay? And in order to answer the ones for the heterozygotes, you know how there's two boxes here that are both 0.16, so you need to do a 2 times 0.16. So if I ask you what is the allele frequency for the heterozygotes, because you recognize there are two boxes, 2 times 0.16 is 0.32. Okay, so that's going to be your allele frequency for that. And then the little r, you only have one box, so you just go with a 0.04. So 4% will have, around 4% you can expect will have little r genotype. 32% will have big R, little r, and then 64% will have big R. And you would expect that because, look, the allele frequency of the big R, the red, far is way more than the white, right? 80. So in this flower population, that means that 80% of the allele frequency is big R. So, 80% of the allele frequency. So that's how frequent the allele is going to come up, 80% of the time. And then 20% of the time is going to be the little r. So hopefully that makes sense to you. You answer this question. Go ahead and pause it if you want to answer, but I'm going to go ahead and tell you right now that the answer to this, if a population is in Hardy-Weinberg equilibrium, that means that the population is not evolving, okay? Not evolving. All right. So what are some mechanisms of evolution? At the most basic, smallest level, evolution means that you're changing the allele frequency of a population, okay? You're changing what alleles are most frequent in a population. So coming back to this one, right? So look, the allele frequency at this given point, 80% big R, 20% little r, or 0.8 and 0.2. Yeah, after their mating happens, if we shift this allele frequency, so let's say, because this is just a probability, but let's say it didn't really happen that way. Let's say it changed to 0.75 and 0.25, right? Just by chance, we've changed the allele frequency, just by the random chance of what flowers pollinate other flowers. The allele frequency changed, and that is evolution. This small, so the flower population evolved from one generation to the next because it changed the allele frequency, okay? So let's take a look at what can cause these small changes in allele frequency is 3 things: genetic drift, gene flow, and natural selection. All right, let's look at genetic drift first. So genetic drift is a random way of shifting gene frequency or allele frequency. And this is just like what we looked at before with our population of red flowers and white flowers. So here's our allele frequencies, 0.7, 0.3, and just randomly, right? The pollen flies from here to there, bees do their job, they pollinate flowers, and because of completely random occurrences, in the next generation, you get a shift of allele frequency. Then randomly again, right, there's some pollination going on, and then the third generation, you get a complete disappearance of the Q of the white flower allele, and 100% becomes the big R. So look, big R, big R, big R, right? We've lost completely that little r. So genetic drift is, I have in big bold words, random, because it shows you that just by random pollination or random chance matings, you can actually shift allele frequencies over generations, and that is a form of evolution. It's a form of changing gene frequencies over time, but it's random, okay? Let's look at the next example. So there's 2 specific examples of genetic drift here. One's called the bottleneck effect. So this is, again, random, right? So let's say it's a bad day, and this bottle represents, and the beads inside represent the allele, the gene pool of a certain population. So the different color beads are the different alleles in a population. Something terrible happens. There's a bottleneck event. This can be where, like, the majority of the individuals are going to be killed. It can be a fire, a forest fire, you know, some kind of horrible flooding event. If you're an aquatic creature, maybe, you know, sewage comes into your pond or lake and just destroys everything. But there's some kind of really awful bottleneck effect. And so the survivors that are surviving, the frequency of the alleles change, what alleles you have also shifts, right? We've completely lost any individual with these yellow alleles. So the surviving population is has a much smaller gene pool than the original population. And that's what a bottleneck does. There's examples of bottlenecks. Cheetahs appear to have experienced at least 2 genetic bottlenecks in the Ice Age, and then also when people hunted them almost to death, right? Almost to extinction. So think about the health of this population, right? So because they went through 2 of these events, right? One event narrows your gene pool down, really makes your gene pool less varied, right? You have less variation in there. If you go through it twice, right, you have a lot less variation. So is it healthy for a population? No, it's not, right? So a healthy population will have varied amounts of alleles, like lots of different alleles, a healthy variation within the gene pool, so that individuals can be different from one another, and so that everybody can have different strengths and different weaknesses, and so they're not going to be wiped out by one catastrophic, like one virus, let's say. But you know, so cheetahs are not as healthy as they were in the past, more than 10,000 years ago, because their genetics are very similar, and so they're going to be more alike now, right? It's like interbreeding. You don't want to interbreed because your gene pool gets narrower and narrower, and then you start having abnormalities show up. Number 2 is something called the founder effect. So it's another genetic drift that's random. However, it changes the allele frequency. So this is where a small group of individuals will colonize an island or something far away and then just start a new colony or population there. So this is an example in 1814. 15 people founded a very small island, a colony. Only one of them had a recessive allele for retinitis pigmentosa, which is a kind of inherited blindness. Out of the 240 descendants, 4 of them have this disorder, 9 are carriers. The frequency of that blind allele is 10 times greater than on the island of Britain. So this is an example of when you have a small gene pool and somebody there has a recessive allele, the likelihood of getting a homozygous recessive person is greater if you have a smaller number of individuals to mate with, right? Because everybody's going to be interbreeding almost, right? And so that recessive allele can get amplified in a homozygous individual just by chance mating more than if you were on the mainland and you had like millions of people to mate with. And that, you know, the recessive allele is not going to show up very often, but in this case it does. So the founder effect also affects gene pools in this way, where you have a very small gene pool, because that's what the founders are. You have a very small population of founders that create that gene pool, and the offspring are actually not that healthy. They have less variation. Okay, and the next one is called gene flow. Gene flow is where genetic exchange is done with another population. It may result in the gain or loss of alleles. Gene flow tends to reduce the genetic differences between populations, making populations more similar. So my examples here are, I think the easiest thing to think about is global travel in humans. So if you think about before ships were invented, before, you know, airplanes, all that stuff, people who evolved, like in Norwegians, would look very distinct, right? They had very distinct features, and their gene pool was isolated from people in China, and they were isolated from people in Africa, right? And they were isolated from people in South America. So everybody had very distinct gene pools. But gene flow is where now we're interbreeding, right? So global travel, we can fly over, people are living in different regions of the world where their ancestors evolved from, and we're interbreeding, and that gene flow is going to reduce the differences, making the populations more similar. So if you think about just all of the individual humans of the planet moving around and sharing gene pools, right, and their alleles will be shared, you know, so now dark skin allele will show up in Norway, right? If a person from Africa were to mate with somebody from Norway, then you would have somebody that is in the intermediate, and then you would introduce the allele to a new gene pool. So basically it can make individuals more similar, okay? Or cross-pollination of plants across hundreds of miles. So the genes are flowing from one population to another. All right, the last, and that's random as well, and then the last process that can cause microevolution to occur is natural selection, which is not random. So that's very important to remember. This is not random. And it's the only process that promotes adaptations. So let's take a look at natural selection. So remember, this is nature selecting for what's out there. So if you had this population of mice, and where very few individuals are light, very few individuals are very, very dark, and then you have kind of this, most of your individuals are going to be in intermediate shade, what does nature favor, right? Because this is the variation of the individuals in that population. But let's say that the white mice are easily seen by night owls, right? Because white shows up in the moonlight and the white mice get eaten, they're disappearing, and you shift. So there's a shift in the favor of the darker mice. That's called a directional selection, right? So the nature is selecting for a direction in one direction, and that's in the direction of dark fur. You might also look at the other two scenarios, right? So this is where the sort of extremes are favored. So maybe the very, very light mice and the very, very dark mice have very good hiding spots. They are able to hide from their owl predator. But for some reason, these middle colored mice are easily picked out. And so you'd get this depression in the bell curve, and you have you'd favor the growth of the white mice and you favor the growth of the dark mice. So that's called disruptive selection. You're basically disrupting the mean or disrupting the average. And then you have stabilizing selection, where you select for that mean. So the dark mice and the light mice, they both show up, the owls eat them, and then in this scenario, right? And so it really favors this intermediate mouse, which is not easily seen by the owl. And so that's a stabilizing selection. So you're going to have to look at, remember that these three graphs are graphs of what is directional selection, what is disruptive selection, what is stabilizing selection, and this is part of the natural selection. All right, so this is a concept check. I'm going to go ahead and give you the answer here. So if the average size wings survive, then you're talking about stabilizing. Okay, that average is favored. All right, so this is sexual dimorphism, sexual selection. So this is the number 4, sexual selection. So it's kind of a subset of natural selection in which individuals with certain traits are more likely than others to obtain mates, right? So this is kind of a funny, but it happens mostly in animals, right? So we have some kind of selection process where one sex will favor traits in the opposite sex that are necessary for producing offspring. But they're not sort of directly related to the environment, other than the individual of the opposite sex wants this trait. So, for example, these finches, sometimes, the males and females will look different, which is called sexual dimorphism. A lot of times the males of animals are going to be the bright, colorful ones because they have to attract the females. The females are usually the more plain colored individuals in an animal pair, because her body is spending more energy on the production of offspring. So there's a lot more machinery that goes on, right? She has to give the food and nutrients to her developing baby and getting fat. And then since he doesn't have that job, his job is to sort of create. So these colors, these pigments are energy suckers, right? They do cost metabolically energy. So the more colorful something is, the more healthy a lot of times that individual is. Flamingos, if you think about flamingos, the more pink they are, it means that they're eating more of their food and they're healthier than the duller, paler flamingos. And then we all know about basically just the different kinds of sexual selection processes. There are fights. You can physically fight for mating, access to mating. You can have, there are spiders and birds that have to do a dance. There are birds that have to build houses to please their mate. There's all kinds of really interesting, funny selective processes that animals have. And then also what's funny is going to like a dance club and watching humans do the same kind of weird mating ritual. Anyway, so that's sexual selection and sexual dimorphism. Here's a question for you. The process of mutation and what create variation in a population? So you can pause it, or I'm going to tell you the answer right now. Mutation and sexual reproduction, letter A. Number 10, you can pause it. What produces adaptations to the environment? This is going to be natural selection, okay? So which I didn't mention before, but an adaptation would be in that, this example here, right? So let's say that the white mice are picked off and we have this directional selection. So you would say that dark fur is an adaptation to their environment, because that adaptation, right, having dark fur is protective and it helps them survive in that environment. So this natural selection is the only one that gives you this adaptation, right? The other ones are random, so you can't really adapt to anything, but in this case, natural selection will produce an adaptation. As a mechanism of evolution, natural selection can be most closely equated with, so pause, or it's answer C, unequal reproductive success. Okay, so this is an example, last example in this chapter where we have natural selection happening right now in human populations, and this is in Africa and parts of Asia, we can see. And we can see that the dots are areas with malaria, and the color is the incidence of sickle cell anemia. So this is kind of interesting because sickle cell anemia is not something that you want. It is a disorder that is very painful for people to have. It can cause death. So in normal circumstances, you would find that the allele frequency for sickle cell is low, right? You don't have a lot of sickle cell. But where you have malaria, that sickle cell allele is actually protective. So if you're a heterozygous individual for sickle cell, you don't get malaria. It's protected. And so in areas where there's a lot of malaria, those dots, right? Deep, deep in malaria country, you have a lot of frequency of the sickle cell. And as you go away from the malaria, so the tips of Africa or more inner Asia, the allele frequency of malaria goes way, way down. Right? So, sorry, the allele frequency for sickle cell goes way, way down because you don't have that pressure of malaria. So you only will see allele frequencies, you'll see allele frequencies fluctuate depending on your environment. And in this case, for humans, the environment includes malaria, which would cause you to have more frequent sickle cell alleles. All right, that's it.